Hello everyone, welcome to Get My Class. So let's solve this question here. It says the DNA dependent DNA polymerases catalyze polymerization in option A 5' to 3', option B 3' to 5', option C A and B, option D 5' to 5'. What are they talking about? They're talking about the DNA dependent DNA polymerase catalyze polymerization. So what are they talking about? They're talking about the DNA polymerase, okay? And this DNA polymerase is what? It is DNA dependent. What do you mean by that? DNA dependent? Because during the formation of, or you can say during synthesis of the DNA, they of course need the support of the template to synthesize the new DNA, right? So here, suppose this is the template 5' to 3', okay? Each single strand acts as a template. We all know that DNA is double-stranded in nature, but here during the formation of or during the synthesis of new strand, the DNA gets separated as single strand, right? And in this particular strand, then this is 5' to 3', this strand is acting as a template. Okay. Now to the template, now this DNA polymerase, we are dependent on the template to synthesize the new strand, because the new strand will be what? The new strand will be complementary. Okay, new strand will be complementary. Whatever the nitrogen bases are there, it will be complementary to that. Okay. This is what? This is newly synthesized DNA. So the polymerase, the DNA polymerase, we call it as DNA dependent because it is dependent on the template, DNA template to synthesize the new DNA. That is why it is called as, this is what? This is polymerase, DNA polymerase. Okay, DNA polymerase. Okay, now during the synthesis, if you see, yes, so during the synthesis, it is going from, if you talk about the synthesis will be from 5' to 3' ends. Okay, this is, and that is why the template it is choosing is like the template is having 5' to 3'. So it will start from the 3' of the template, but its direction, moving direction will be from 5' to 3', because it can add nucleotides only at the 3' end. So this is 5'. So here, if you have to cut here, so this will become the 3' end. So like this, it will continuously synthesize it. Okay. But then one important point, so DNA polymerase cannot directly add the nucleotides here in the beginning, and that's why they make use of something called as primer. RNA primer is added here, and this RNA primer will provide that free 3' hydroxy end, so as to add the nucleotides further by DNA polymerase. So what is the direction of DNA polymerase? It is 5' to 3'. 5' to 3'. Okay. Now, so option A says 5' to 3'. That is absolutely right. Option B says 3' to 5', which is wrong. Option C says A and B, which is again wrong. Option D says 5' to 5', which is totally wrong because there is no existence of 5' to 5'. There is always 5' to 3' or 3' to 5' with respect to the DNA double helix, because they are anti-parallel. But when we're talking about the movement of the enzyme here, that is DNA polymerase, it will move from 5' to 3'. Okay, so let us conclude the answer. What is the answer? The answer is option A, that is 5' to 3'. I hope you understood the explanation. Thank you.